Alright, I want to show you sterile technique, tube-to-tube transfer. Right here we have a slant with bacteria growing on it. It is Enterobacter aerogenes. We have another one with Pseudomonas fluorescens. We have a third one with Serratia marcescens. We then have 3 tubes of what's called phenol red broth. This is phenol red broth. It has 5 grams of sucrose per liter. It also has an inverted tube inside called a Durham tube. The way this works is that you inject it with bacteria or inoculate it with bacteria. If the bacteria ferments the sucrose and produces acid, we get an acidic environment. It changes the phenol red from red to yellow. If, in addition, then gas is produced in the fermentation process, this tube then will get gas and will get a bubble inside indicating that gas was produced in the fermentation process. Now what I want to show you is sterile technique. Here we have our flame. Our flame is basically creating a sterile environment. It grabs bacteria, pulls it in, bacteria go up, they die, they come in, they get burned, they die, and it creates an area around it that is sterile. So I want to work within that area. Now I want to hold everything in my hands. So I'm going to hold these 2 tubes in my hands. I'm going to hold my loop in my hands. I want to be able to take the lids off with my fingers. I want to flame the tops of both of those lids. I then want to go ahead and flame my loop. Okay, my loop is hot, so I'm going to take it in and just basically dab it a little bit there. Grab some bacteria, put the bacteria in, shake it all around. Okay, flame the tops of my loops, put my lids back on, and then flame my loop to end. Alright, I'm going to repeat that exact same process now with this tube and this tube with this bacteria and this bacteria respectively. Alright, so 48 hours have passed. We have incubated all of these for 48 hours. Here is our Enterobacter aerogenes. We can see here yellow tube indicating fermentation, acid production, and see that big old giant gas bubble. Here is our Pseudomonas fluorescens. Right here we can see no fermentation producing acid, right? No bubble inside that Durham tube. Here is our Serratia marcescens. In this case, we had acid production from fermentation of the sucrose, but no gas production because of different fermentative pathway. And right here, a teeny bit of gas in there, but that's not enough to be a positive for a Durham tube test. All right, so we're now ready to do a plate to a slant transfer. This is a slant of Simmons citrate agar. Simmons citrate agar has citrate as its sole carbon source and ammonium ion as its sole nitrogen source. The pH indicator is bromothiamyl blue, which is blue when the pH is greater than 7.6 and green when the pH is less than 6.9. When no other carbon source is available, some organisms utilize citrate as the carbon source that ends up releasing CO2 through metabolism. The CO2 combines with Na+ and H2O to produce alkaline compounds like sodium carbonate. These compounds increase the pH, making it more basic, which then turns the initial green to blue. So what I want to do then is I basically want to take a plate. This is Citrobacter fruendii. It has been streaked for isolated colonies. We can see up here nice isolated colonies on this streak plate. I want to take one of these isolated colonies then, and then just take my loop and just spread it out across this slanted area here, just in a nice S fashion across the slant, using a sterile technique. Again, my flame is keeping things sterile in an area around it, so I want to do this all within this flame. I don't want to contaminate my plate, so you notice I'll just lift my plate up a little bit, and then I'll also end up getting my tube ready to go. So I'm going to start by flaming my loop. Okay. I will then cool my loop on the edge of the plate. I will then just take a little bit from this nice isolated colony right there. I will then take my lid off, hold the lid in my hand, flame the loop here. Take this down then and just gently across this in a nice S fashion. Just go right up that tube. You'll then flame the tip again, close this lid off, flame my loop to finish this off. Okay, and set my loop down here to cool down. Okay, so now 48 hours have passed since I inoculated this Simmons citrate agar. Here's my tube of Simmons citrate agar. We can see now that the green has turned to blue. Basically, it has become more alkaline, and that alkaline then caused the bromothiamyl blue to change from green to blue. All right, what I want to demonstrate then is what's called a stab technique. I have my culture here, it's on a slant, it's of Enterobacter aerogenes. I'm going to then go ahead and do a stab into a tube of Sim medium. Sim medium then is a semi-solid medium because it takes half the agar, so this is really soft. And what it allows for is it allows for bacteria to move through the agar, and you can actually look and see if the bacteria are motile or not. So we use a needle in this case instead of a loop because the needle has to go straight down into the Sim medium. We want to make sure that it is nice and straight. So I'll go here then, we will heat this loop up. While it's cooling down, I'm going to grab my two tubes here. I'm going to then take the lids off of my two tubes. We will flame the two tubes. I'm then going to go down into the Sim medium here, and I'm going to get bacteria right off the surface. And then what I want to do is I want to stab straight down nice and carefully, and straight out nice and carefully. Flame my tubes, put on my caps, and then I am done, except for flaming my needle. Okay, so 48 hours have passed. This has been incubated. This is the Sim medium that we just inoculated. It's hard to actually see on this, but basically what we're looking for is a stab straight down, and then we're looking for cloudiness going around the outside. So if you go, you hold up the light, you look at the cloudiness, and you can see that this is a motile bacterium because it's all cloudy throughout. If this only had a stab and growth only along that single stab, then it would be non-motile, but this one then has growth throughout all the way in the tube, so it is motile. Alright, so now I want to show you the quadrant streak technique. Right here I have a culture. This culture is a mixed culture of Serratia marcescens and Bacillus subtilis. The pink in here is the Serratia marcescens, Bacillus subtilis is a white bacterium. These then are mixed there. My goal is to try to get them separated so that I have nice isolated colonies of my Bacillus subtilis and my Serratia marcescens. So I basically want to take it, I'm going to do a little streak here, I'm going to flame it, do another streak to the side, flame it, do another streak, and then finally flame it. And I'll do a continuous streak in the open area, trying to only get a single bacterium on my plate to grow into a nice isolated colony. So we'll start by flaming the loop here. Okay. I'll take my tube and my lid, we shall put it there. We shall go in then and get a nice colony. We'll flame it, put that down. I'll open this lid barely, and I'll just barely do a nice little streak here on this side. Okay, I'm going to flame my loop again. I'm going to turn my plate to the side. I'm going to take this and I'm going to drag this through in a continuous motion from that original culture, nice and neat. I'm going to flame my loop again. We'll turn the plate again. We'll cool it off a little bit. And I'm going to take it through that area that I just did, nice and neatly. Flame my loop one more time to kill all the bacteria. Turn my plate, cool my loop, and then I'm going to continuously streak throughout the whole area that hasn't been touched yet by those bacteria. Okay, flame my loop, and then I will incubate this plate upside down for 48 hours. Okay, so 48 hours have passed, and we can look at the plate. So if we look at this plate, right, we will notice that there's a lot of interesting stuff here. We have red colonies and we have white colonies. Most of these colonies are kind of a mixture of red and white. Serratia marcescens then is the red, Bacillus subtilis is the white. Now on here there are isolated colonies what appear to be isolated, but the reason that I chose Serratia and Bacillus is that Serratia is pretty cleany, so it's hard to get really good isolated colonies. So here's a really nice isolated Bacillus colony. Here's another nice isolated Bacillus colony. We have a pretty good isolated Serratia marcescens colony right there. So overall this is pretty good. We then went from thicker to thinner to where we finally got individual bacteria on the plate. Alright, so now what I want to do is I want to make a smear preparation and then gram stain a colony of my Serratia marcescens, the pink one, and a colony of my Bacillus subtilis, the white one. First thing I want to do is I want to take my bottle here, put a teeny tiny drop of water on the slide, and we will put a teeny tiny drop of water on this slide as well. Okay, we'll go ahead and light the Bunsen burner. Alright, now I'll take my loop and flame it. And I'll open up the lid, cool it down a teeny bit on this side. Take a little bit, that colony right there. We'll swirl it around in a nice big circle here. To allow it to dry. Reflame my loop. Now Bacillus is a little tougher, but there's one nice little colony right there. You just want to touch it only, and we will swirl it around. Nice and big for my Bacillus. And I'll flame my loop. First step is done. Now I have to wait for this and this to dry before I heat fix it. Alright, so a couple minutes have passed and we now have dry samples, and so I want to heat fix it. Just pass this over 1/1000, 1/1000, and this one is heat fixed. And we'll grab this one and do the same. We just go 1/1000, 1/1000, and this one's all ready to go. Alright, now I want to go ahead and stain these. So I'll just move all this stuff out of the way really fast. Even off that, move this out of the way fast. Put these two over and we'll put them on the staining tray. Then we will add our various stains to all of these. So gram stain procedure starts with crystal violet. So I'm going to take my crystal violet, we'll just put a drop of crystal violet on my slide, and we'll cover my smear in crystal violet. Okay. Now we have to wait for 25 seconds while that crystal violet is on that slide. What crystal violet does is that crystal violet, you can see, is a purple stain. That purple stain then is going to stain the bacteria purple. Gram-positive and gram-negative bacteria will both be purple at this stage of the staining process. Okay, 25 seconds have passed, so here we'll take this and we will just gently wash off the crystal violet from both of my smears. right? Now that the crystal violet is off, I want to add my secondary stain, which is going to be grams iodine. So for grams iodine, then I want to put the drops right on the smear. Okay, and again, then I want to leave this for 60 seconds. So what happens is that the grams iodine then goes in and combines with the crystal violet to make a grams iodine crystal violet complex. Gram-positive cell walls then have a thick layer of peptidoglycan. Gram-positive cell walls then will hold that crystal violet grams iodine complex in that peptidoglycan. Gram-negative cell walls then have an inner layer or an outer layer of a phospholipid bilayer, and they have a small inner layer of peptidoglycan. So for those, then they're not going to be held as solidly in that peptidoglycan, and I'm going to be able to do what is basically called a decolorizer step using ethyl alcohol. Okay, so a minute has passed, and we shall wash off our grams iodine. And we shall wash off our grams iodine. Make sure that we wash it really well, because we don't want any of that left over. We want to make sure that our decolorizing step works very well on this. Okay? Decolorizer then is basically an ethanol solution, ethanol and acetone. We'll put decolorizer on and make sure that we have plenty of decolorizer on there. Okay, that decolorizer then needs to stay on there for 30 seconds. Now what the decolorizer does is that basically it's going to wash the purple or the crystal violet grams iodine complex out of the gram-negative bacteria, and it will leave in the crystal violet grams iodine complex in the gram-positive bacteria. Okay, so then what we want to do is wait 30 seconds. It's here, and so now we're going to take this, we're going to wash off this decolorizer really well here. Okay, we'll wash off the decolorizer really well here. Make sure that we don't have any decolorizer left on there, because we want to counter stain with safranin. So final step then is safranin. Safranin is pink now, which is a contrasted color to the blue. So we're going to leave that on for 25 seconds. Now this is called the counter stain. Counter stain means that it looks different than the blue, right? The blue in our crystal violet, grams iodine, gram positive then stays in. The pink is going to be on top of it, so they'll remain blue, and the blue kind of overpowers the pink. However, in our gram negative, it was colorless, right? At this point where we used our decolorizer. Now we add our counter stain. The counter stain turns it pink, and so our gram positives should be blue, and our gram negatives should be pink at the end of this stage. All right, we'll wash this off really well. And once we're done washing it off, the last step is to gently blot it. We want to make sure that we gently blot it, because we don't want to get rid of anything that we shouldn't get rid of. So we just want to tap it, not rub it, so that that stain does not actually leave that slide. I can rub the bottom, but I don't want to rub the top. And then we'll just gently blot it. Okay, I'll get rid of everything off the bottom. And there we go. Now we're ready to move on to the microscope. So here is the smear from the red colony on the plate. We're assuming this is Serratia marcescens, but we'll have to do a gram stain to make sure. So we gram stained that colony, and this is under 4x. We'll increase magnification to 10x and refocus it. We'll increase that magnification to 40x, Increase the light coming in, and we'll focus it. Okay, right now these are looking pink, so that would indicate that they are gram negative, but sometimes you have to make sure under oil. So we'll go here all the way up. Put a drop of oil on the slide. Wheel in the 100x objective. And we'll go ahead and focus that. And here we go. So we can see these. These are definitely nice ones. I won't tell you what they are, but definitely pink. We can see the shape of those, and we can see whether they are gram negative or gram positive, and that looks great. Okay. Alright, so very clear what those are and what the gram stain qualities are. And here's our second smear from the streak plate. This was one of the white colonies, supposedly Bacillus subtilis, but we have to double check with our gram stain and microscopic observations, and we want to see how pure it is. So here we are under the 4x, shoot up to the 10x, focus, shoot up to the 40x, focus. We'll increase that light to increase that resolution in there, and we'll easily go between the 40 and the 100x, and we will put a drop of oil right on the slide, wheel in the 100x objective, focus, and there we go. Alright, pretty clear if it's gram positive or gram negative, easy to see the arrangements.